So first, in the light-dependent reaction, we have the photoactivation of photosystem 2. Two photons of light are absorbed by a special chlorophyll molecule, and the electrons within the chlorophyll become excited. This is photoactivation. Each chlorophyll loses two electrons that each reduce plastocanoid. Then we have photolysis, where the lost four electrons from photosystem 2 must be replaced. So each chlorophyll is now a powerful oxidizer. So it oxidizes water to take its electrons back. This puts the hydrogen ions into the gradient, and it also releases oxygen as a byproduct. Then we have the electron transport chain. The excited electrons jump from chlorophyll A to plastocanoid, and then to other electron carriers which pump hydrogen ions into the gradient, and then finally it arrives at its destination of plastocyanidine. The proton gradient is created by the photolysis of water and the pumping of hydrogen ions from the ETC, as well as from the ETC of NADPH as well. Chemiosmosis, the H ions passively move through the membrane from high to low concentrations, so they move from the thylakoid space out into the stroma. And then photoactivation of photosystem 1. Two photons of light are absorbed by a special chlorophyll molecule. Electrons within the chlorophyll become excited, which is yet again photoactivation. And then each chlorophyll loses two electrons that reduce ferredoxin. The ferredoxin takes four electrons. And then NADPH formation, electrons from ferredoxin are transferred into reductase to reduce NADP plus into NADPH using hydrogen ions from the stroma. And then finally, photophosphorylation, which using the energy from the hydrogen ions passing through the gradient, they are able to convert ADP into ATP using the energy of sunlight obtained from photosystem 2. Cyclic phosphorylation. This happens when sometimes there is not enough ATP or the supply of NADP plus runs out, so ferredoxin will donate its electrons back to the ETC instead of to NADP reductase, which allows for more pumping of hydrogen ions into the thylakoid space. And the final products of this reaction are ATP, NADPH, as well as oxygen. So the next reaction there is the light-independent reaction. And so the first phase of this is carbon fixation, which occurs in the top part of it, where RUBP is fixed using carbon dioxide and this happens in the stroma by the enzyme Rubisco. This reaction does not require any sort of ATP or outside energy. Next, we have RUBP and Rubisco. RUBP is a 5-carbon molecule that gets fixed by the enzyme Rubisco. It adds a carbon dioxide to the molecule, which turns it from a 5-carbon molecule into a 6-carbon molecule temporarily. And Rubisco is the enzyme that does this fixation. Next, the 6-carbon molecule is split into two 3-carbon molecules, which are called glycerate 3-phosphate. The molecule occurs when the 6-carbon molecule created by carbon fixation is split into two 3-carbon molecules. This molecule does not have a 2-to-1 ratio of hydrogen to oxygen. Next, we have reduction. Because the molecule does not have a 2-to-1 ratio of hydrogen to oxygen, it has to have a hydrogen added to it. So ATP and NADPH are used to add a hydrogen ion to glycerate 3-phosphate, where NADPH supplies the atom and ATP supplies the energy, which after this it creates triose phosphate. And then once you get triose phosphate, in order to resupply the cycle with RUBP, most of the triose phosphate that will be made will be regenerated into RUBP. This means that for every 6 triose phosphate that are made, 5 of them will be regenerated into RUBP to continue the cycle, and only 1 of them will be made into glucose. And then after the reduction phase, we have the regeneration phase. Triose phosphate is regenerated into RUBP using ATP, where one of the phosphates from ATP is donated from ATP to the end of triose phosphate. And then what would happen if the amount of RUBP, glycerate, 3-phosphate, and triose if the lights were turned off? After the lights are turned off, all the products from the light-dependent reaction will run out and be gone. So this means that RUBP and triose, the levels of those will fall because both of these require ATP, which comes from the light-dependent reaction, while the levels of 3GP, glycerate, 3-phosphate, will rise because in order to make this molecule, it requires no ATP or energy. It just requires RUBP and the fixation of carbon, not any outside energy or ATP.